All right. Hello, everybody. I'm still Peter Dearden from Genomics Aotearoa, and thank you for joining us for a fantastic seminar today. As I always say, if you want to be on our mailing list, then feel free to send us an email at genomics.aotearoa@otago.ac.nz, and we'll put you on the list and let you know of all the other fantastic seminars that are coming up. But none of them will outshine the seminar we're having today. So today we have the fantastic Maren Wellenreuther, who is from Plant and Food Research and the University of Auckland, who's going to talk about inversions as facilitators of eco-evolutionary change, as you can see on the screen, which sounds incredibly exciting. And Maren's going to talk a little bit about her journey to this topic, and so I'm going to let her get on with it in a second. If you could turn off your cameras while she's talking, that would be useful, and feel free to put questions in the chat as we go along or at the end, and hopefully we'll have some time for questions at the end. So please feel free to, if you've got any burning questions, either bung them in the chat or talk to us afterwards. Maren, it's all over to you. Off you go.
Great. Well, thank you, Peter, for an introduction. Yeah, as you've all heard, my name is Maren Wellenreuther. My talk today is entitled Inversions as Facilitators of Eco-Evolutionary Change. So my talk today will be structured roughly in the following way. I will, like Peter said, give a little bit of an introduction just to tell you a bit about my background. And then I will also provide a little bit of background about structural genomic variants, just to sort of lay the scene and make sure that we're all on the same page. And then I will dive into two case studies that I have been working on. The first is I'm going to use the Australasian snapper that many of you are familiar with, a native species in New Zealand, to look at the question, how common are structural genomic variants? And after that, I will use a different study system that I have been working on for a number of years. It's a bit of a less charismatic species. It's a seaweed fly, Coelopa frigida. And this is a really great study system to look in more detail at the role of structural variants. And here I will look at inversions and ask the question, what are actually the impacts of inversions on sort of eco-evolutionary dynamics in a species, sort of looking at fitness consequences. Okay, so that's roughly sort of the outline of my talk today. So I will dive straight into it. A little bit about myself. So you can already hear that I'm talking with a bit of an accent. So I am from Germany. I was born in the middle of Germany, in a beautiful town called Göttingen. It is a very old, 1,000-year-old university town. This is where I started to study biology towards a master's degree. And I realized while I was studying that quite early on that I actually had an interest in marine science, marine biology. So I moved from Göttingen up north in Germany to Hamburg, another old university town, and I started to study fisheries science. And I got to spend a lot of my time on these really cool European research vessels, like you see on the top right-hand side of the slide. Here's the Walter Herwig 3, and I got to spend lots of time on these European research vessels, surveying fish populations and analyzing different things. Then, because I always like to travel and adventures, I actually finished my master's degree not in Hamburg, but instead I moved to Australia and I studied at the University of Adelaide. And I also moved a little bit my focus from fisheries science towards fish ecology. And I got to study this beautiful fish here that you see, the magpie perch, Chelodactylus nigropies, and I looked at its foraging behavior. And as you just heard, I've always been curious and adventurous. So after my master's, I thought, the grass is always greener on the other side, and how wonderful would it be to study insects? So I moved to the other side of the world. Oh, actually, I did my PhD first. Actually, I didn't move to the other side of the world. I just moved to New Zealand. And I actually dived a little bit deeper into fish ecology and also started to work on evolutionary aspects for the first time. And I worked on triple fins, and many people in New Zealand refer to them as cockabullies. They're found all around the coastline. They're New Zealand's most abundant reef fish. And as I said, I started to combine evolutionary theory with ecology. And then actually, after sort of having spent almost 10 years on marine aspects, marine biology, I thought, how wonderful would it be to work on insects? And I went all the way to Sweden to do that. So here you see me actually during my first postdoc, but then also my second postdoc, first working on Calypteryx damselflies, and then later on in Schnurrer damselflies, looking at sexual selection and color polymorphisms and the maintenance of color polymorphisms. Super fascinating, I loved it. I really dived more and more into genetics at that stage of my career, and then really in 2013 started to develop quite a keen interest on structural genomic variation and maintenance of structural genomic variants. And I started to work on seaweed flies, and this is something I'm going to talk to today about as well. So I also took on a faculty role at Lund University and worked there for a number of years. And then, well, I'm here. I actually got a job offer at Plant and Food Research in New Zealand, and I say back to the roots because after having worked then on insects for 8 years, I came back to New Zealand, and I now oversee a group called Seafood Production at Plant and Food Research in Nelson. So that's anything related to aquaculture and fisheries. But I should say that I still have a very active interest in things outside of fish. So I also still work on damselflies and seaweed flies. So it's a little bit about myself, just to give you a bit of a flavor about my background. And so now I would like to dive into the first part of my talk that really sort of seeks to provide a bit of a background about structural genomic variation and why I think this is a really fascinating topic to be working in. So to begin with, I think if we are talking about genetic variants, the sort of familiar picture that we are usually thinking of when we are thinking of genetic variants is this. It's sort of the raw data that we're often dealing with. You know, we're all familiar with this. I call it the A's, the T's, C's and the G's, and some N's usually as well. And most of the time when we look at this sort of data, we actually end up converting it into sort of matrices or data sets that contain these single nucleotide polymorphisms, SNPs. And that's great. I mean, there are many really good things about SNPs. One of the most important things about SNPs is they're easy to deal with and they're very common. So they're often very evenly and densely spread across the genome. This is a really positive thing about them and why we should be using SNPs. So if you look at the human genome, for example, we can say they roughly occur every 300 base pairs, depends of course, but just as a rough estimate. So this gives you really a good number of genetic variants to work with per human genome, say at least 11 million markers, depending on filtering and thresholds and all of that. And having this high number of markers that is sort of segregating across genomes is a really important feature because it makes them really amenable for GWAS analysis, looking at LD, QTL analysis, you know, selection scans, all of that. They're good markers for all of that. But one of the questions that I've been sort of really interested in is sort of just to look into, if we're looking at the whole genome and we are interested into the parts of our favorite model species, you know, that are under selection, what parts of the genome are actually selected on, and are SNPs the right thing to use? Because when we actually look at the whole genome and the whole extent of variation that we find in the genome, we realize that the genome differs in quite dramatic ways in many instances. We know that structural variation, and I've defined this here in blue, what I mean by structural variation. So this is genomic variation between individuals that can affect the presence, the abundance, the position, and or direction of a nucleotide sequences. These structural genomic variants, we know from human studies, are very common, and they also occur across the genome. And so in my work around 2000, structural genomic variants, and sort of asking myself, if I really want to understand what is happening to a species in nature, then I probably have to sort of expand my view and expand it beyond SNPs and encapsulate the full extent, ideally, or more of the genetic variation that is segregating in the species of interest. If we look at sort of a bit of a full picture of genetic variants, we can look at it like this. So we can roughly sort of structure it from sort of quite simple genetic variants all the way to quite complex structural changes that can occur in the genome. So single nucleotide polymorphisms at the top, but then we looking at copy number variants, so insertions, deletions, duplications, which can be extremely common in genomes, human genome, but also other genomes. We're also talking about things like changes in the chromosomal location when we're talking about structural variants. And here I'm really talking about translocations, fusions, fissions. These have been extremely important also in human evolution and are now increasingly being found in other species as well. We're also talking about change in the orientation of nucleotide sequences, such as inversions. And I will talk a lot more about inversions in the later part of my talk. And of course, when we're talking about structural variation, we're also talking about changes to whole sets of chromosomes, such as aneuploidy, polyploidy. So these are all structural genomic variants. And so I'm sort of trying in my research to keep an open mind a little bit, or trying to sort of think a bit beyond the SNPs and trying to capture the full extent of genomic variation that is segregating. So when I talk about structural genomic variation, that's what I'm referring to. And just to briefly sort of sum this up, this part, if we should realize, I guess, that genetic variation, we have a whole size spectrum of genetic variants when we are looking at a genome of a species that really ranges from single nucleotide polymorphisms all the way to whole changes in the sets of chromosomes. So the size spectrum is enormous. When we look at structural variants, we also typically differentiate between balanced structural variants. So there we're just talking about change in the position or the order, or we're talking about unbalanced structural variants. And here we're talking about gains and losses of nucleotide sequences. So these balanced variants like inversions, but also translocations. And then when we're talking about these unbalanced structural variants, we're really talking about these insertions, deletions, duplications, things like that. So this is just to keep that in mind and keep that sort of overview ready now as I'm going through the case studies. So in the next part, I really want to sort of talk about, now that we've sort of looked a little bit into the types of genetic variants that we actually have in the genome, I want to ask the question, if you're just looking at a non-model species like snapper, can we ask the question, how common are these different types of genetic variants actually? So this is my first case study, and I just want to say a few things about the species. So of course, many of you are very familiar with snapper. It's a native species, Tamure, in New Zealand. It's of quite significant recreational importance in New Zealand. It's sort of considered by many people as a Rolls-Royce of inshore fisheries species, but it is also of quite significant commercial importance as an inshore fisheries species, and of course, massive cultural significance. Maori have been fishing snapper for hundreds of years. It's a really sort of quite iconic species in New Zealand. So when we started to look into the sort of types of genetic variants that are maybe important in snapper, one of the first things we did is that we assembled the genome of the species, and that was maybe in around about 2016. So using slightly sort of more old-fashioned methods, as we would say now. And here I just have briefly cataloged the way we have gone about assembling the genome. So you can see we used a range of short and long read sort of libraries. And then we used ALPath for the initial assembly, ended up with roughly 6,000 scaffolds. We used some optical mapping to join things, creating super scaffolds. We added a linkage map to it, chromonoma, and we ended up with these 24 chromosomes that you can see in the figure here. You see the linkage groups on the inside circle. On the outside circle, you see the different contexts and scaffolds that are sort of aligning to that. So this is was really good to have in place. And then we were going out into nature, and we were sampling snapper from 3 locations in the country. So here you can see it, the Hauraki Gulf, the Manuka Harbor, and Tasman Bay. And from each of these locations, we collected 2 males and 2 females. So 4 individuals per location, 12 in total, obviously, and we sequenced them at 30x whole genome. And we used these genomes to look at the genetic variants that are present in snapper. What did we find? When we were looking at the genomic data, we could analyze or identify the number of SNPs that we found in the species. And here you can see we found around 6 million SNPs in the snapper genome. And we also found structural genomic variants, a little bit over 20,000. And on the left, you can see a circus plot that shows the different 3 different types of structural variants segregating in the snapper genome. So you can see inversions in blue. This is the inside circle. You can see deletions in the middle circle, and then duplications in the outside circle, and the different linkage groups, of course, or chromosomes indicated on the outside. You can see that these structural variants are found across the genome in each linkage group or chromosome. What was quite interesting as well is when we actually looked at the number of base pairs that were impacted in the snapper genome by the different types of genetic variants, we found that structural variants impacted 3 times as many base pairs as SNPs did. So that kind of gives you a bit of an indication that these structural variants, because they are more rare, but they are bigger, they actually have quite an influence on the genome. Next, we ask the question, how are these structural variants, not just partitioned across the genome, but also what is their size distribution like? And here again, we are focusing on the 3 common types. We're looking at duplications, deletions, and inversions, always indicated in the same color as before. So inversions in blue, deletions in red, and duplications in black. And you can see that the majority of them are very small. And actually the mean indel length was 4.8 base pairs. So this is quite an important finding, because if you're familiar with the literature on structural genomic variants, you'll see that most studies filter out these small variants. They actually just get rid of that, as so they're looking often at 100 kb or something like that, or 10 kb minimum, and disregard anything less. So here it shows you that filtering decisions are really important, especially if you want to understand the full extent of genomic variation that is segregating in your species of interest. In the next step, we actually ask, are these structural variants, given that they are so having a large impact on the in terms of base pairs in the genome, is there any evidence that they also have an impact on the phenotype? And this is a really indirect analysis now, or way to look at it, but I'm going to talk about it very briefly, because what we looked at is how many of these structural variants are actually intersecting partially or fully with genes. And we could see that actually that structural variants intersected with almost 1/3 of all of the annotated snapper genes. So it's quite a lot. And we also found that when we looked at sort of upper and lower 1% quantiles of Tajima D and pi, so evidence for selection or regions that are under selection in the genome, we found that a sizable portion of structural variants were located in those regions. So both of these quite indirect measures, but they're at least suggestive that these structural variants have probably an impact on the phenotype and thus also on the evolution and fitness of the species. Okay, just to briefly sum this part up, because I want to move on to my last sort of part, which is a bit longer. I want to just say that, first of all, you saw that while SNPs are the most common structural genomic variant in the SNP genome, structural genomic variation actually impacts a higher number of base pairs in the SNP genome, 3 times as many base pairs. Also, several of them intersected with genes and are in sort of regions that are, you know, under putative selection. So this suggests that it probably has an impact on the phenotype and evolution. But I really wanted to move on to this other case study, the seaweed flies, you know, to really sort of look at the question, what is the impact of structural genomic variation on fitness? And in order to look at that, it's actually much better to work on these seaweed flies for many reasons. They are much more amenable to ask sort of detailed questions about fitness consequences. Now we'll talk a bit about that. So a little bit first about inversions, because the rest of my talk will really just now focus on inversions, now that we've established that structural genomic variants are important and that they are common in many genomes. So if we're looking at inversions, the first thing I want to sort of explain is what is an inversion and what are the consequences of having an inversion. So an inversion is actually a part of the chromosome that breaks at 2 parts, flips around 180 degrees, and then gets reinserted back into that chromosome. So it's an inverted region. And these inversions have immediate impacts on the genome, and they also have less immediate impacts on the genome. They occur usually during meiosis, and often we know that they can be triggered by environmental factors such as radiation, chemicals, viruses. Also repetitive elements are often associated with breakpoints. We also know that other inversions make the 3D structure of chromosomes a little bit more amenable to breakage. So, but the important thing about inversions, what I want to have you think about, is that they have immediate impacts on the individual and also less immediate impacts. So here I just show you again, it gets reinserted back into the chromosome. So you've got this inverted sequence here. Okay, and they have immediate and less immediate impacts. If we look first at the immediate impacts, so one of the things is obvious: if an inversion happens and the breakpoints sit within a gene, then often that inversion is lethal. So it's an immediate impact, and it probably won't be maintained in the population. But the other immediate impact is that many inversions immediately have something what we call a position variegation effect, and this means it has an impact on the expression of the sequences that are, or the genes that are inside the inverted region, just due to the change in the position, because it moves genes closer or further away from regions that are, for example, heterochromatic regions or things like that. So depending also on the size of the inversion and how large the spatial differences then are, you have immediate impacts based on the expression. But a really important thing about inversion, and this is probably also the reason why they have been studied so much in the last years, is that they have actually quite a strong impact on the evolutionary trajectory of species. So if we're thinking about an individual having an inverted and a non-inverted part of their chromosome, so their heterokaryotypes, so they're carrying both, then you can look at it, you can look at these different arrangements as they're acting like pseudo-populations of haplotypes. And here the alleles are differently colored, and you can see standard arrangement, inverted arrangement. What is happening is that we have the typical sort of selective processes that are always happening on the genome. But if you have an inversion, and you have an individual carrying the inverted and non-inverted part, then that means that in that region, you do not have recombination. So the recombination is suppressed. And because of that, because you have a lack of recombination in heterokaryotypes, you also are dealing with drift in these regions because you have a different effective population size of that genomic region. And so if you think about it, the genome is always exposed to mutation, selection, recombination, drift, but in the inverted region, recombination and drift behave differently. And so these regions evolve differently from the rest of the genome. And that's really important. So these are the non-immediate impacts of inversions on species that are carrying them and where they are segregating polymorphic in the population. They have been actually described in a number of study species, and here you can see some really famous examples, such as the white-throated sparrow, ruff, Anopheles, Drosophila, Temema walking sticks. They've been really well described. And why is that? Because inversions often capture massive chunks of DNA. All these inversions that have been well described are large. They're large inversions, and they can be roughly categorized into 3 different types. And you see that here on the slide. So a lot of inversions that have been described in these model species for eco-evolutionary studies, let's say, you can see good examples where inversions are having an impact on the behavior of the species, such as seen in rainbow trout, where you have the anadromous and resident ecotypes, for instance. Or inversions commonly impact mating systems, which has been well described in the white-throated sparrow or the ruff. Or they can impact phenotype-genotype associations. That's also really well described, for instance, in heliconius butterflies or Temema walking sticks. So these are roughly the 3 categories that have been really well studied when it comes to inversions. And when you are looking actually at these species that have these inversion polymorphisms, they're really kind of, depending on where they are at with regards to evolution, they span the whole spectrum of variation. And I will run you through some of these examples here. So here, for instance, when we look at the zebra finch, you can find examples where inversions actually, here we are dealing with a stabilized within population polymorphism that still has similar frequencies, small effect sizes, things like that. Going to mosquitoes, so Anopheles, here we get frequency shifts caused by inversions associated with clinal latitudinal variation. We can look at honeybees and the Atlantic cod. Now here inversions are implicated to cause altitudinal variation and cause the alternate fixation between these different ecotypes. Looking at monkey flowers, here we're looking at locally adapted, these life history shifts that are caused by inversions. Looking at the European corn borer, now we're going more towards really gene flow is sort of more and more, comes to a stop. Here we are finding that inversions are actually associated with QTLs that are directly having an impact on reproductive isolation. Same here with Drummond's rockcress, and all the way to the alternate fixation, in Helianthus sunflowers. So this is really, inversions have been implicated to be associated with a whole spectrum of genetic variation. And I've been studying inversions really using the seaweed fly, Coelopa frigida, for a lot of my work, and they're fantastic. They're a really cool system that has been heavily worked on already in the 1950s and 1960s. Roger Budlin, who some of you I'm sure are very familiar with, did his PhD on seaweed flies, and they have this really cool inversion polymorphism on chromosome 1 that actually impacts 11% of the genome. It has around 1,000 genes in it. It's massive. And not only that, it has a really strong impact on the phenotype. So you see here some flies, and these are males, and you can see the inversion is called either alpha or beta. So you have the alpha form or the beta form. And if you have homokaryotypes that are carrying both of the alpha forms, they are 3 times as large as the homokaryotypes that are carrying the beta form. And the heterokaryotypes, so the individuals that are carrying the alpha form and the beta form, they are intermediate. So we're talking 9 millimeters, 6 millimeters, 3 millimeters. So massive size range in males, not in females. So sex-specific effects. So there's a massive inversion. You can see it by eye in males, and it has a really big impact. Not only that, they're a fantastic model in the lab. They have a generation time at 25 degrees that takes 2 weeks for the whole life cycle. And there was an allozyme marker available for determining the inversion, the alpha and beta inversion. I had to learn allozyme staining during that time, which was quite challenging, but I managed. But they're not only a fantastic model for the lab. I wrote here related to Drosophila, but much more interesting. I hope I'm not offending anyone with that. They're not just good for the lab, they're also fantastic for the field because they occupy a really wide environmental gradient. You can catch them easily with just a little butterfly net that you see me holding there. And they're extremely tolerant to environmental extremes. So you can see salinity and temperature gradient is massive. So great model for the lab, great model for the field, super cool inversion polymorphism, has massive effects on the phenotype. So this is a great model to really understand the impacts of carrying this inversion on the eco-evolutionary dynamics in the system. What is maintaining it? What's going on? And so we looked at the literature. We saw that this fly was occupying clinal gradients in Europe that was established by Butlin, but also by lots of other people. And so one of the first works I did actually was that I used a reciprocal transplant experiment in 2017 to look at if they actually show evidence for local adaptation, and they did. So we published that, but then we were really asking what is actually going on. Like, they show clear evidence of clines, they show clear evidence of adaptation. Let's do a big genomic study to actually sort of better understand of how that is happening and what is happening. So to do that, we actually started first out to develop some good resources. So we assembled the genome, and not only that, we developed a SNP marker for the inversion, because that allozyme marker, it worked really beautifully and really great, but it is much easier to work with a SNP marker. So we developed a SNP marker, and we did this genome assembly, which you can see here. I won't go into the details, but just to say, it's a really nice genome, long reads combined with a linkage map. It's a small genome, 180 MB. It was pretty straightforward and easy. So we're having a fantastic genome to work with. We've got a SNP marker, and then we set out to do sort of a larger scale population genomic study to better understand the dynamics of this inversion in nature. So we actually, had a postdoc, I still have her, Claire Moreau, in Quebec in Canada, and she did this crazy field trip over 3 weeks. She sampled 8,000 kilometers of coastline and sampled populations of Coelopa frigida, so in North America. So all of what was known about Coelopa frigida came from Europe. So we went for the first time to North America, looking at a parallel cline to Europe and saying, do we get similar patterns like we found in Europe, and why is that? So you can see here, she sampled a massive environmental gradient. And the way we were sampling it, we did 20 populations along this gradient. In each population, we collected 100 flies. You can see massive latitudinal gradient. And whenever we came, they're called seaweed flies because they live in decomposing seaweed. These wreck beds, they're called wreck beds, these decomposing beds of seaweed. These wreck beds, they differ in size and seaweed composition and temperature and all of that. So we measured all of these ecological parameters as well. And of course, then we used the SNP marker to then also look at inversion frequencies along this cline and how that relates to the environment and how much parallelism we can find with Europe. So we looked at this. Here you can see the different inversions that we've documented in North America from our field trip. First of all, I should say we didn't find any clinal variation, nothing that clinally varied with temperature or salinity, but we found massive heterogeneity between populations. We looked a bit more into this and we found that actually there is definitely some extent of continental parallelism. We found that there are conserved features between North America and Europe, such as the alleles and their effects on size is exactly the same. So we have these large males, intermediate males, small males, dominating these wreck beds. And we can also see that there is some relatedness of the inversions with the types of wreck beds that they're found in. So for instance, wreck bed depth and composition was a strong predictor of inversion frequency. So a bit of a different story compared to Europe, but definitely some conserved features. The inversion impacts on the phenotype are conserved, and also wreck bed characteristics were able to determine the dynamics that we were seeing. Now, I want to talk about one last study that we did. That's only 2-3 more slides. And this is really kind of showcasing a little bit the cool stuff that you can do with flies, I think. Not just like a massive field sampling like we did here, but also then taking them into the lab and simulating what may be happening in nature. So what we did next is that we really did some experimental evolution on the flies to better understand why we're getting this quite, heterogeneous pattern of inversion frequencies in North America. So the experimental evolution assay that is depicted here on this slide, the really cool thing is that it allowed us to disentangle the reproduction stage from the survival stage. So we disentangled that so we could really understand the fitness components that were coming from either natural and sexual selective processes. And the way that we did that, you can see we have these different, generations in the lab. And so we wanted at each generation, we wanted to assess the role of reproduction, so sexual selection. But we did this by putting adults on a pile of virgin seaweed. So with clean seaweed, we left them there for 20 hours. We allowed them to deposit eggs. And then the next morning, we collected all the eggs and we preserved the adults. And then some of the eggs that we didn't need for preservation, we moved on to a fresh set of seaweed. So 1,000 eggs, we always moved on to the next generation. And then we let them develop, and we collected the emerging adults. We preserved them for subsequent genotyping. So this way, we were able to look at then the frequency of the inversion and the proportions of the 3 inversion karyotypes, both by looking at the adults that emerge, but also by genotyping the eggs. And this way, we were able to really look into, we had non-overlapping generations, and we could look at the contributions or the impacts of having the inversion or not having the inversion on reproduction, but also survival and development time and things like that. And what we found is that actually these 3 inversions, they correspond to 3 different life history strategies. And we did a lot of modeling as well, but just in a nutshell to show you what we found is that the alpha-alpha homokaryotypes, so those individuals that carried the alpha inversion both times, they're the very large males, right? So they are large. They show very slow development. they are long-lived, and they have a very massive mating advantage, because mating in the species is forced. The males jump on the back of the females, they hold on to them with all of their. We can also look at the beta-beta males, so the ones that are carrying the other, so the beta inversion. Here we are having a very small size. They are very fast at developing, and they have low reproductive success. But they're really, when we look at these different types, is that we actually find that the large ones, they have a real advantage in these high resource environments, these really long-lived wreck beds, these thick wreck beds. They're deep, they're warm, there's heaps of substrate, there is no risk of being washed away. Whereas these small males, they were found in these tiny wreck beds. And you know, when the tide comes in with the moon, they get washed away sometimes once a month, or you have a storm, they get washed away. So these were these really fast, sneaky ones that could just develop super fast. They were the first ones to mate with a female because the other ones hadn't emerged yet. So in the intermediate ones, there's a massive heterozygote advantage we found in the species. They probably have less genetic load. They overall do better. And in particular, lab experiments have shown that in high density conditions, they have a significant advantage. So this is really then also aiding the maintenance of the inversion in the population. Okay, so not to go too much over time here, just to quickly sum up. I hope I was able to show you in my talk today that structural variants have a large impact, or can have a large impact, on the genome. I hope I have been able to show you that they actually can have also an impact on the phenotype and fitness, and ways to look into that, ways you can study that. And I probably just want to say, like, it's a real challenge these days now, I think, for all of us to maybe do like a good job to discover the full extent of structural variation that is segregating in animals, or at least more of what we're doing right now, to better understand the role of these structural genomic variants in evolution. And with this, I'd like to say thank you.
So I'd just like to thank Maren again for an excellent, fascinating talk, which included large quantities of insects, which always makes a talk good. So I'm very pleased. Yeah, if you, as I say, if you're not on our mailing list, please send an email. And I think Jess put our email address in the chat right at the start. So send an email and we'll put you on the list for more awesome talks like that. So thanks again, Maren. I'll do the clapping. And,
thank you, everyone.
Thank you.
Thanks, Peter.
And we'll see you all again soon.
Excellent. Have a good weekend, everyone. Bye-bye.